Hi everyone, Kevin here. We're back with the end of module number 4. Let's pick it up where we left off. And that was finishing up topic 3. So we'll finish up topic 3 and hit topics 4 and 5 in this video. Remember in topic 3 we talked about all these influences on fermentation and fermentation rate. And I want to emphasize that in this class, we deal with two things. We deal with both the chemistry of wine and we deal with the microbiology of wine. And with microbiology, we're dealing with living organisms. Therefore, we deal with biochemistry. Biochemistry is the chemistry of life. So those living organisms are important to wine, and we have to look at their biochemistry. So we'll be dealing with both topics. If you go through this list of influences on fermentation, temperature affects both chemistry and microbiology, primarily microbiology, because as temperatures increase, all reactions tend to go faster, including those in the microbes, including the biochemical reaction of the microbes. So chemistry and microbiology are both affected by temperature. pH affects both chemistry and microbiology. pH affects the chemistry of the wine itself, and it affects the fermentation rate of those microbes. Sulfur dioxide has impacts on chemistry. It changes some of the chemical reactions in the wine, and it also impacts microbes because it inhibits microbes. Nitrogen, same thing, impacts the chemistry and the microbiology. More importantly, the microbiology because it's a key nutrient for yeast during fermentation. Sugar obviously impacts chemistry, impacts the microbiology. It's the source of food for our microbes. Alcohol impacts chemistry. It obviously impacts the chemistry of the wine long after fermentation is done. The amount of alcohol impacts the mouthfeel of the wine and how the wine tastes to us. But increasing alcohol levels also inhibit microbes, and we talked about you can kill yeast if the alcohol percentage gets high enough. And we'll talk specifically about the impacts of alcohol in module number 5. And finally, oxygen impacts the chemical reactions in the wine, both during fermentation, but primarily during aging. That's where we limit the amount of oxygen. Oxygen impacts microbes as well, and we talked about the impact on yeast cell membranes in that last lecture. So, as we go through the class, think in terms of the chemistry of the wine and the microbiology of the wine. Let's move on to native yeast fermentations. Native yeast fermentations are a growing trend in the industry, and it simply means we're allowing the native yeasts that are stuck to the outside of the grapes in the vineyard, we're allowing those yeasts to do the fermentation. So wineries love this because it's a natural process, so we can use it in our marketing. The people who like natural wines, natural processes, and in other words, like the least amount of additives in their wine, they love the fact that we allow native yeast from the vineyard to do the fermentation. Note too that when we allow these native yeasts that are stuck to the grapes to start the fermentation, they often don't finish the fermentation. In any winery, there are a ton of yeasts in the winery itself, in the air, on the walls, in the tanks, depending on how well you clean the tanks. There's a ton of yeast around, and oftentimes what finishes the fermentation is some commercial strain of yeast that may or may not have been stuck to the grapes when the grapes came in from the vineyard. And there's a link here at the bottom of this slide to a Wine Business Monthly article that talks about that. It talks about some research done in Oregon and how no matter what yeast you started with, what finishes the fermentation is typically a commercial yeast, the same kind of commercial yeast that winemakers often add to a fermentation. So even if we don't add yeast, there's enough yeast in the winery and in the air, etc., that the commercial yeast will often finish the fermentation, because those are the ones that are tolerant of high alcohol levels. Here's a little research that a research project separate from the one linked in the last slide that shows what I just talked about. And if you look at this slide, you'll see that there are 4 lines on here that represent the relative growth, or the cell density, the cell count of 4 strains of yeast. And they're shown at the top here. So 3 of those, Clecera apiculata, Candida stellata, and I forget what the M stands for, but the M, it's Mechnicovia pulcherrima. Those 3 are considered wild yeasts. They're yeast that we often don't add to our fermentation, but they're common in the vineyard. And you can see on day 1 of fermentation in the graph shown that those 3 have relatively high cell counts. Look at Saccharomyces cerevisiae, our typical wine yeast. It basically had 0 or a very low cell count. So our 3 so-called wild yeasts start at a fairly high cell count, and they grow along. Cell counts remain until, in the case of the Clecera apiculata, on day 5 they rapidly die off. And that's because Clecera apiculata is very sensitive to alcohol levels. As fermentation goes along, it takes a few days for alcohol levels start to go up, and as they go up to 5–8%, something like that, yeast strains that are less tolerant are going to die off. And that's true for the Clecera apiculata, and the Mechnicovia, the red line, also dies off at about the same alcohol level. The Candida stellata is a little more tolerant, so it goes out to about day 7, day 8, and then it starts to die off too as the alcohol gets too high. But then look what finishes fermentation. It's the blue line. It's Saccharomyces cerevisiae, and its cell counts stay nice and high all the way to the end of fermentation, because typically Saccharomyces cerevisiae is tolerant of alcohol. And whether the Saccharomyces cerevisiae cells came in on the grapes from the vineyard or were just present in the winery, either way, as the wild yeast die off, the Saccharomyces cerevisiae takes over and can finish fermentation. So this is very typical. What's cool about having 4 yeast strains participating in your fermentation is they all give different flavors. So when those 3 wild yeasts are fermenting for the first 5 to 7 days, they're producing certain flavors, and those will add to the complexity of your wine. If you did not do a native fermentation, what you would do would be you'd add sulfites right at crush to kill off the native yeast. Native yeast are often sensitive to sulfites, and so you can reduce the populations of the native yeast dramatically by adding SO2 at crush. In that case, you would add yeast, and Saccharomyces cerevisiae would have a high cell count from the beginning of fermentation and would go through and finish your fermentation. But that means you only have 1 species doing fermentation and less flavor differences from yeast. So wild yeast fermentations tend to be more complex. There's a risk that your fermentation could produce bad flavors, because maybe one of these wild yeasts produces bad flavors, so it's risky. Winemakers tend to get to know vineyards, though, from year to year, and the yeast strains tend to repeat, though some current research is suggesting that it's not as repeatable as we thought from year to year. But in general, winemakers get confident that wild yeast from a certain vineyard are not going to produce off flavors, and then they have more confidence that year after year they can do a native yeast fermentation and you won't get bad flavors. You'll get more complexity, which is really nice. You just have to live with a little risk of off flavors. So that's a native yeast fermentation. Last topic in this module is what's the difference between yeast and malolactic fermentation. So we talked about them a little bit already. Yeast fermentation, of course, is also known as alcoholic fermentation. We also call it primary fermentation. That's what turns our grape juice into wine. Malolactic fermentation is also called secondary fermentation. That's what turns malic acid into lactic acid. And that's optional. Some wines go through it, some don't. And some of what I just said is summarized here, but the key point in the difference is that yeast do the alcoholic fermentation, bacteria do the malolactic fermentation. So you never say, I added yeast to do malolactic fermentation, nor do you say, I added bacteria to do my alcoholic fermentation. Yeast does the alcoholic, malolactic fermentation is done by bacteria. So both are single-cell organisms, as you probably know. Yeast are eukaryotes, which means their cell structure, their internal structure of the cells, is similar to other eukaryotes like us. It means that they have organelles within the cells that are surrounded by membranes. Prokaryotes, of which bacteria are one, they don't have separate organelles within the outer cell membrane. So they're single cell organisms, an outer membrane, but no separate organelles within the cell. Eukaryotes have the outer membrane, but individual organelles within the cell, each of which is surrounded by its own membrane. Eukaryotes tend to be larger. They have a cell nucleus. In some ways, maybe more complex biochemistry. Actually, in many ways, more complex biochemistry than prokaryotes. But prokaryotes have a lot of the same biochemistry as eukaryotes, but they don't have those internal organelles, and they don't do some of the complex biochemistry. Here's a look at those two cells. Prokaryotic cell. It shows you the plasma membrane, the cell membrane on the outside of the cell in both cases. And in the case of a eukaryote, the DNA is actually in the nucleus, so you have a separate nucleus with a nuclear membrane around it. In a prokaryote, though, the DNA is just free in the cell. It can aggregate like this is shown, but it's not in a separate membrane-bound organelle. Ribosomes are what convert DNA to protein. They read the DNA and produce proteins, and those are found in both cells. But in eukaryotic cell, you can see that there are a lot of different organelles. There's a mitochondria there. This is the Golgi apparatus shown in blue here, and there are other organelles shown as well. Where in the prokaryotic cell on the left, it's basically just the plasma membrane, the cytoplasm, the cytoplasm inside, and then everything else is just in that mix in the middle of the cell. We'll be looking at yeast biochemistry in particular as we go along in this class. That's it for Module 4, just one video. Thanks for watching, and we'll see you in the next module.